morning, class, and welcome back to our discussion on transcription. Next lecture will move into translation, but before we get there, I want to talk about gene expression. I kind of left you guys on the last question of our last lecture to think about how can the expression of genes be regulated, or how can transcription be regulated. So when we think about an organism, whether it's a single-celled organism like a bacteria or all the way up to something as complex as a bird or ourselves, many different genes need to be turned off and turned on in each individual differentiated cell, whether it's a heart cell, liver cell, muscle cell, sex cell, germline cell, all these different things. And also, even within the single cell, if you're a bacteria or something, you don't always need so much of a protein or less of a protein. Life needs to be efficient because efficiency is going to allow us to live, although it's not the most efficient, but more efficient than any computer we've made. So gene expression and how genes are turned off and turned on and the amount of proteins made from a gene is all something that needs to be controlled. We've talked about certain control systems already in the way of feedback loops, specifically in-product inhibition, where we have a metabolic pathway ran by enzymes, and the final product of the pathway then in turn becomes an allosteric inhibitor for the first product. So that would be one way I think transcription would work. But we're going to cover a few different ones today. We're going to talk about DNA methylation. I'll probably mention acetylation, although that one's not one that's too big for you guys, so we won't get into it. It's histone modifications. And finally, we'll talk about suppressors and enhancers, proteins that can sit on the DNA genome and either silence a gene or slow down production or speed up production. And we're going to use a bacteria to explain that one. So, not too long on notes today, and as you guys learned, I'm probably not in class, so you're having to watch this somewhere else thanks to our wonderful NYP program that I have to go do today. So. DNA methylation is a big one that I want you to kind of understand. And what happens is that a methyl group is going to be added to the 5' end of a cysteine DNA. You don't need to know it that much. Just know that it's going to stop transcription. And so what happens is in your DNA, a little cap basically on all of the C's. It happens in places where you have CGs. Because C is complementary to G, these little caps get put on with a methyl group. And a methyl group's nothing more than a carbon and some hydrogens attached to it. It's like a full grouping of hydrogens. CH4 is a meth. In this case, it would be C3 because the carbon would be attached to something else as well. But basically, that methyl group makes it so when you have these regions of DNA that have these caps of methylated caps on them, it doesn't allow, especially in the promoter region, it doesn't allow RNA synthase to attach to it. And so when RNA synthase can't attach to it, the gene does not get produced. Okay? And so this is a very important mechanism for cells because it's used to differentiate cells. We find it in almost all types of life. We see methylation. Normally it's on the C's. There's a few types that end up on the A's, just not near as common, so we won't talk about those. And really just in all life except for a few archaea bacteria, pretty prevalent in mammals. It's really common in plants. Plants have huge genomes. I mean, they can have, 6-ploid, meaning they have 6 sets of chromosomes for each thing. And so they have lots of methylation. We as humans have quite a bit of methylation. And so, like I said, it's used to differentiate cells. It's also very important in, sometimes, we have 2 copies of every gene, right? I mean, different alleles, you might have different versions of it on 2 chromosomes. But sometimes you don't want to use both of those chromosomes. It would make too much of a protein. So on certain genes, you actually methylate one of the chromosome genes so that you only make so much of the protein and keep in a healthy amount. So methylation, really, really important. But it can also be caused by reactions to the environment. So it's number one is really important to differentiate into different cells to make sure certain areas aren't turned on and turned off. Something called histone modification does this as well. But then methylation, we've found, also occurs with reaction to the environment. So when we first really started to learn that DNA was the hereditable thing, it's all in your genes. Everything comes from your genes. We thought, environment had very little to do with it. The more we learn and the more we get going, we find out the environment has a really big role in playing how your genes are expressed. I want to continue talking about this a little bit for the purpose that most methylation during a sperm cell, so when the sperm and egg meet, most of the methylation patterns are actually deleted during that time. So shouldn't be passed on from parent to child because the sperm gets wiped of all methylation patterns, and so does the egg. And then the ones that are important are put back on by methylase enzymes. And it happens there, and it also happens during the production of sex cells, like when cells go through meiosis to create sperm or egg, it should be wiped too. So there's a double place where they should be wiped. And so normally these things wouldn't be passed on from generation to generation. But we have found in recent time that occasionally parts of it do get passed on from mother to child or father to child through the sperm. And so this is another way of inheritance, that you can inherit methylated patterns on your DNA. And that can cause great concern, because if the wrong methylated patterns come, you can end up with some interesting things. And we've even found evidence that methylated patterns can be passed on from grandparents, which means probably even further back in time. So it's really leading to this idea that environment plays a much bigger role than just genetics, although it's still based in genetics because it's the methylation patterns there. But so far we have found no pattern with how these methylated patterns are passed on and not deleted during sperm production. So there's a few instances as you could study more, but just something interesting to think about. How does the environment affect you and your offspring, and therefore the fitness of a species, because life is really all about your offspring and the next offspring of those offspring, because that is how life continues on. Now in a more spiritual realm and everything else, that could be different today. So another interesting one is the methylation of the X chromosome. Some of you in class heard this, some of you probably didn't. Hopefully you know that female mammals have two X chromosomes, typically have two X chromosomes. There can be interesting things, like there are men who have two X chromosomes with certain weird crossing over events that cause them to get an SRY gene and they still grow boy parts. So chromosomes don't exactly tell sex, but we'll get into that later. But most females have two X chromosomes. In most cells, one of those chromosomes is actually inactivated. Okay, and DNA methylation, so like pretty much everything on this chromosome gets methylated, and it turns into what we call a bar body. So one of the chromosomes is just actually turned off. It's just X'd out. It sits in the cell, but it's just never used for transcription. It's never used for anything. And an interesting thing is it's completely random which one gets turned off and on, or at least to our understanding, it's random. And so in female mammals, in most cells, only one X chromosome is actually doing any work. And then there's a little bit of the X chromosome that has some genes on it. This would be the equivalent to what's actually on the Y gene on males. So think, if I'm looking at this little chromosome here, maybe just this little section down here, that would be the equivalent of the Y chromosome. Those get turned on, the rest is all turned off. And you can actually see this in cats, which is really kind of interesting. So in certain types of cats, the color of black and then this orangish color that we see in cats is actually on the X chromosome. So if you're a male cat, you get one chromosome. You either get black or orange, so you will have black male cats and orange male cats. But if you are a female who happens to get, say, a black one from mom and an orange one from dad, so a black female cat breeds with an orange male cat, the orange has to get passed on the X, and what we end up getting is this kind of thing we call a chimera of a cat, or what's the other word of this? Calico is what the name of the cats are. Chimera means we get kind of both chromosomes. And so what happens is, depending on what chromosome is turned on in different areas of the cat, you get different colors. So Over here, we have our male and our black male. Doesn't matter. Female, if they're homozygous for each trait, will get that. But if they do have this heterozygous orange and black, you end up with different parts of the cat that turned into different things. So this little ear section, that part, the X chromosome that was black got turned off, and over here the X chromosome that was orange got turned off. So kind of an interesting thing and pretty unique in females. Can also cause some issues if you have genetic mutations, but just something I think most people don't realize. You always think, oh, I got 2 X chromosomes, and you do, but only one of them's ever actually really working full time. And we don't know which one in each place. So just a cool little biology thing. And methylation, just to talk about how it shuts down parts of the chromosome, just like you'd shut down parts of genes that you wouldn't use for a certain type of cell. Back to this epigenetics is what we're going to talk about next, and how it relates to methylation. Something Ivy just wants you to ponder and think about, and I think it's really important to think about too. And epigenetics is the heritable changes to the gene expression of genes without changing the DNA. So this is things that could be passed on. So when I talked about there are methylation patterns that do get passed on from the sperm and the egg, these are heritable then, and they're changes or epigenetics, things that the environment can actually change the way your genome is expressed. So your genes do not change, but how your genome is expressed can change. So this can be caused by nature. Things like that we know of that cause methylation patterns to change are stress, the types of food we eat, the toxins we eat. Now, we always focus on the negative, but we actually find that if you have more happy thoughts, you actually change your genome to express things more happily too. So I think we shouldn't always focus on these negative things that can change your genome, but a lot of good things can change your genome too. And just because there's toxins around and changes your genome, we think of that as bad, and toxins probably are bad, but maybe that change in the genome actually helps us adapt to those toxins and therefore survive. It's survival is much different than being happy or anything else. So when we think about evolutionary, the human body and our genes and how they've evolved, we can't always put it in today's time. We have to think of the past with our psychological state and everything else. Stress probably helped us survive a long time, and those changes helped survive. But now that we know about them, can we change them mentally? So could you possibly, this goes back to my idea of conscious evolution of DNA now, have we evolved the point where I can mentally think, try to control my brain and see the more positive sides of things, and therefore change my epigenetics, change my methylation patterns to actually make sure that my children might even see things more happily? That is a crazy idea. I should study that more. But all kinds of things might change it. So these are just some things that we have found can change your epigenetics, or not necessarily their epigenetics, but at least we know that they change your methylation patterns. And if they change your methylation patterns, it is possible they can be passed on to your young. Whether they are or not, we don't know. Exercise can change your body's expression in great ways. I think I mentioned to some of you, we have found that methylation patterns will actually cause people who exercise a lot to have their cells and the most of their body use fat instead of sugar during exercise, which can be a bonus thing for you. And then their mind actually uses the glucose. So all the sugar goes to your mind so you can have clear thought, while other things go to help your body run, which is pretty awesome. We know that drugs, it doesn't have to just be of abuse, but drugs in period definitely change methylation patterns even after first use. Alternative, anything we come in contact with is probably doing something to our genome, and so much to study here, but we won't go far. I do want to talk about one case study. You might have actually done this one in psychology too, and it's just an example of epigenetics. It's also an example of how environment can actually control genomes. In plants, you see this all the time. Plants are freaking amazing, and I wish we had more time to go into them. If you want, you should look up some phenotypic plasticities of plants. We'll talk about that a little more in evolution, but it's kind of on the same idea. But we do have lots of studies with mice, and we can look at their genome with mice. And I'm not going to get into this really detailed, like what is the ABN and everything else. But what we found is mice, which are mammals, very similar to us, when they're babies, they need to be close to each other. So these little mice are close to each other. Picture them in their little, hidden behind your wall, and they're all huddled up together as little babies, and they're suckling on mama's teats to get to their nourishment, and they get touched a lot. And this touch actually makes sure that they increase the expression of certain genes, which actually slows down methylation of a certain gene, and they produce lots of this GR gene. And that's how a normal mouse should turn out. We have found if we keep the mice separated, so we don't let them, they're born, we still feed them, but they don't get touch. They don't get like that rubbing and the touching. And babies, human babies are the same way. If it's untouched, we have serious, serious problems. And what we find then is we don't express this one gene, which causes more methylation to occur. And a certain gene gets turned off because of this. And we find that mice that aren't touched as babies, exact same genome, these are all clones of each other, end up yellow. So by not being touched, their actually genome expression is changed to be yellow, very unhealthy, not good. And it's all because they just weren't touched as babies and held. And so mammals, especially a lot of mammals, us, mice, pigs, actually require some kind of like almost massage or something, the touch, I would call human touch in humans, to actually develop proper brains, we found out. And in mice, it even changes the color of them. So definitely shows that environment does have a big role. It is not all about the DNA, although it is still about the DNA because the DNA is what got turned off. That is all I'm going to get into on epigenetics. I hope it interested you. I wish we could spend a whole year on it. I mean, I wish we could just study it, honestly. It's such an interesting line. But really, that's just an overview. And the last thing we're going to talk about is how proteins can also affect gene expression. And we're still talking about everything that would affect transcription. So all of this inhibits or would increase transcription to change things around. So things that also will change gene expression or regulate gene expression are things called transcription factors. So if you remember back to the only enzyme you have to know for transcription is RNA polymerase, and it was this really big molecule. There's also things called transcription factors that bind to this RNA polymerase. And most of these transcription factors are going to cause it to increase the speed of transcription or to make transcription more likely to happen, which would increase the speed of it or the rate of how much protein is made. And so there's tons of different transcription factors, and there are transcription factors for every type of gene almost too. So this is just, there's a class of transcription factors, and then there's ones unique for those genes, which means there's a gene to build the transcription factor. It just starts to blow your mind. But these things just bind, and they help to increase or decrease the speed of transcription or translation. And then we also have activators and suppressor proteins. So activator proteins, they're going to bind to the DNA, and they can actually enhance transcription. So they normally don't actually bind to the gene. They'll bind somewhere else on the DNA, and sometimes they cause the DNA to bend right, which makes it more likely for these transcription factors and RNA polymerase to find the promoter sequence and start. So they actually not always right on the DNA, and sometimes they are on the gene right where it is. And we also have suppressor proteins, which kind of do the same thing, and they're going to bind to the DNA and actually silence the gene. So just different ways that our body can regulate when a gene is turned off and on. I don't know how that just happened. But these, more unlike methylation, are less permanent, so these can definitely change quickly, and they change due to concentrations of chemicals within the cell. And I am going to give you guys an example of this in a bacteria, and we're going to talk about the lac operon, lac standing for lactose. In bacteria, unlike eukaryotes, there are no introns and exons. Everything gets turned into mRNA. But bacteria typically arrange their genes so they actually make multiple proteins within one gene. So you have a promoter, and so when one gene here gets made, and there's three of them, lac Z, lac Y, lac A, that's actually alpha, beta, and zeta. They make three different proteins, as you can see here. One makes lactase, this is just lactase, it breaks down lactose. And then we have one that helps transport lactose into the cell, and then we don't know what this one does, but it probably has some very important thing. So all of these are needed for the bacteria to use lactose as an energy source. And so this is just kind of the gene you have. This is the LAC1 gene. You have a promoter sequence. It's going to bring in that RNA polymerase, and then it'll run and make this whole thing. So all along this little thing, we can control when we need to make this as a bacteria. So if a bacteria has lots of glucose, it really is much more efficient to use glucose. So it should not want to make any of these proteins. Why spend the energy to make these proteins if I don't need them? If the bacteria doesn't need them, it would be inefficient, and it'll just go using glucose to do cellular respiration, although in this case it's more like just glycolysis. But if there's no glucose present, then it might want to use lactose. So in that case, if lactose is present, it's going to make these genes and use that lactose that it'll then turn into glucose for energy. But if there's glucose present, you don't want to make it. And if there's no lactose, you don't want to make it either. So this is a really interesting way this one works. So when would a bacteria want to use lactose as a source of energy? And really the only time is when no glucose is present, because glucose is the best source of energy. But if it is not present, it needs to make glucose. So if lactose is present, then it would. So two conditions need to be met for the bacteria to actually turn on its lac operon. And those conditions are we can't have any glucose and we must have lactose present, because trying to make those proteins when both of these conditions are not met would just be a waste of energy. Okay. And so basically the transcription of the lac operon is controlled by glucose levels and lactose levels in the cell. Glucose levels are actually going to be slightly indirect. So similar to like when we measure CO2 to do the rate of respiration in a mealworm, that's kind of indirect, but a byproduct of it is what we're using, and that's kind of what this lac operon uses. So in this thing, we have the gene. We have this thing called a capsite. And it's just a DNA sequence where this little thing sticks onto here. And this is actually an activator protein. So when this is attached to the capsite, we actually increase the rate of this RNA polymerase hooking on, and we're going to actually cause it to start transcription. We also have a repressor protein. So if this is attached, it's going to block the production of the gene or transcription. So first part, when glucose is present, something called cyclic AMP, adenosine monophosphate, okay, so similar to ADP, ATP. In the process, when glucose is present, cyclic AMP levels are very low, okay? And I don't care that you know what these are. You can almost just think of this as glucose. That's kind of this indirect thing. But when glucose is low, cyclic AMP levels are really, really high. So when glucose is low, cyclic AMP levels are high, and in order for the cap protein, this promoter protein, to attach to the promoter region and increase the RNA polymerase action, it must be bound to cyclic AMP, or picture it's bound to glucose. So actually it's when glucose is low, so it's not. So when glucose is low, cyclic AMP binds to this cap protein, and that's going to increase transcription. So awesome, it turns the gene on, it's ready to go. Okay? So that only occurs when glucose levels are low. If glucose levels are high, there's no cyclic AMP to bind to this, and these two do not connect. So cyclic AMP has to be bound to the cap protein to promote it. Okay? Lactose is the other thing that's important. So in lactose's case, we have this repressor protein, okay? And when there is no lactose present, the repressor protein is bound to this. And when this is bound, RNA polymerase can't run, the gene can't work. Okay? If lactose is present, it's going to bind to this repressor, which is going to cause it to release. So they're kind of the opposite reactions if you think about it. So in this case, something binds and it causes it to go and attach. In this case, lactose binds, it causes it to fall off. And so now we can make the gene. But these two have to work together. I'll let you guys read those for a minute. Okay, they have to work together. So the only way we actually will make the lactose gene is we have to have low levels of glucose, which is going to cause the cap protein to start RNA polymerase. And then we also have to have lactose present so that the repressor protein can fall off. And so this is the only way that the lactose gene will actually be made, because if we have high glucose, we're not going to get that starter thing, and it's just not going to run. Okay, if we have no lactose available, even if our glucose is gone, so here's high glucose, it's unavailable, that's that repressor protein stops it. Opposite, we have low glucose, but we have high glucose, it just doesn't go. So hopefully you can kind of look through those and see that that is how genes can regulate. I don't expect you to understand that completely. I'll ask you a few questions, but more I just want you to have an idea of what activators and suppressor proteins might play in the role of regulating gene expression. And you would find similar things in humans, and that is good for today. I'll throw a few lecture questions up there. Hopefully that didn't take you guys too long. Next week we will jump into translation and the actual building of proteins. Pretty unique stuff. And then we will head into a little review of genetics and then off to human body systems and animal body systems. And so close, we are getting to the end of your IB biology career. Don't forget your IAs.